We're gonna get started in a minute. There. Screen. Share. And then get to this to here. And maybe this will work. Yeah. There. And they're good.
They're good. Yeah, if at one point in class, you want to pause and have them ask each other a question to do the things, or I can do it now if you like.
Yeah, go ahead.
Okay.
Ask them about their homework.
How did guys' homework go?
How did the homework go? Yeah?
Okay, before I walk out of the room, I'm gonna do your attendance cards. So, actually, you can turn to somebody you don't normally sit with or right at your table. Your attendance card question of the day is, that asks you about, you can have an imaginary pet. Imaginary pet. What would that imaginary pet be? I should ask like a head space question. But you can ask. All right, so pull out your homework. All right. So, what was the answer to number one? Where's your homework? Okay, what did I do? Your dog ate your homework? I didn't say that. It was funny too. It is funny. It is funny. Who said? But I was just catching fun. It doesn't, well, that's. So the dog, the dog actually ate my brother's homework once. And he tried to tell my parents that, and they didn't believe him, because he got marked off for not bringing his homework in. And then, it kind of showed up at the other end, and they finally believed him. Okay, so what was the answer? Yeah. It's really all right. And, but what's the extra part about an allele? Yeah, the idea, keep in mind with the alleles, the idea that it could be a little different from one person to the next, okay? Because that'll all play down. Let's go ahead. Sharon, what did you get for compound heterozygote?
Individual who has two different abnormal alleles of the same gene. One on a pair.
Exactly, and so that's why it's important to know what allele is, okay? To know that that's the version on one chromosome versus the one on the other chromosome. Got your cell phone. All right, so we can skip back to an autosome. Away from this table.
any chromosome other than a sex chromosome.
Yeah, too easy, too easy. All right, let's skip down to codominant. We didn't really talk about that this year, but we talked about it a lot last year. Codominant.
Equal expression of two different alleles of a gene.
Equal expression, yeah, but you can have equal expression. Oh, this is getting into the weeds. You can have equal expression, but it's, of two alleles. But yeah, and at the same time, but it just means that they both have, a phenotype. Rather than one, than a dominant versus recessive. Codominant means that they both, the phenotype of both exists, so AB blood type, right? So the expression of one doesn't mask the expression of the other. Okay, make sense? I'm gonna cheat on your paper. Okay, so then what about copy number variants? We talked about this a little bit yesterday when we were talking about ultra rapid metabolizers. Laura, I'll let you pick somebody from your table to answer.
I'll pick somebody,
Page. The number of copies of a specific segment of DNA that varies among individuals' genomes.
Right, right. So we talked about CYP2D6 and how sometimes some people have 6 and you can have up to 13 copies on a single chromosome. And if all of those copies are being expressed, then you're making a whole lot of that enzyme, right? And that's how you end up with ultra rapid metabolizers. All right, Aiden, how about histones?
Proteins that DNA wrapped around common chromatid.
Mm-hmm. And what's the important characteristic of those histones?
What do you mean that they open up?
Yeah, they open up with transcription happens, but what kind of amino acids are typically on the tail ends of those histones? That we talk.
start and stop codon.
No, that should be part of it, but what about the amino acids? Lysine, right? And lysine is positively charged because, what's the charge on DNA? Negative, right? So that's why it's important, that important characteristic. And so when we talk about histone acetylation, right? That is changing the charges on those lysines that are hanging out from those histones. See how everything's going to build on each other? Okay? All right. Thank you, Michael. Oh, so hopefully you guys got pharmacogenetics and pharmacogenomics and the difference between those. We spent a bit of time on that. All right, we haven't gone through this yet, but maybe you've had it in PK so far. What about phase 1 enzymes? Yes?
Phase 1 is like oxidizing reduction and hydrolysis, and then phase 2 is usually like a conjugation.
Okay, so what are those two things used for then? Anybody else? Yes,
Phase 1 enzymes we can use to create metabolites, that's what you find a lot of times in the liver. And then the phase 2 enzymes are used to attach large groups to metabolites for excretion.
right. So remember that a lot of times the phase 2 enzymes are important to make those metabolites into a form that they can be excreted one way or another. All right. And so we're going to talk a little bit more of point mutations. I haven't heard from Tommy's table back there, Ella. Point mutations.
A single base pair changing.
Yep, pretty simple, but the most common type, those single point mutations, right? The single nucleotide polymorphisms that we're going to talk about today. And so then we talked a lot yesterday about the definition of a polymorphism. Right? 1% of the population. Okay. And so we just talked about SNPs as well. Cytochrome P450s. I have heard from almost every table. I think I've heard from every table. Anybody else want to talk about cytochrome P450s? What are those? First off, are they phase 1 or phase 2 enzymes?
Phase 1.
Phase 1. So therefore, what do they do? Yeah, they metabolize a lot of the drugs that you guys are going to be, dispensing. All right, thank you all. So that's a good study sheet for you. All right, so we talked about other variations, the insertions and deletions. The other key thing about indels is if they are, they're within an intron, sometimes it affects the efficiency of splicing. If they are in a coding sequence in an exon, if they're a number that is not 3 or divisible by 3, these indels can change the reading frame. And we talked a lot last year about reading frames and how important that is. What is a characteristic of the two different reading frames that are not the open reading frame? So if we have the open reading frame is the one that will give us a sequence for the protein that's going to be made, right? So what happens if we now insert or delete 1 or 2 bases? You get a different protein, but what happens a lot of times in those other 2 reading frames?
They move up more, so we stop it.
Right. So there's a lot of stop codons in those other 2 reading frames. Part of that, it prevents the cell from wasting energy making an incorrect protein. But also it makes a shorter protein. Sometimes it makes it fold incorrectly so that it's ubiquitinated. So the net result can be just less protein. You have the amount of protein that's made off of the normal allele, but the mutated allele with an indel, sometimes you will not make that much protein. You'll just end up with less protein around. And sometimes that matters and sometimes it doesn't. Okay. And then we talked about copy number variations, like CYP2D6 up to 13, and then large deletions. Remember I said GSTM1, about 50% of the European American population is missing that gene altogether from either chromosome. Okay, but there's enough redundancy in the functions of the other. You remember what GSTs stand for? Nobody? Glutathione S transferases. So there, if you're missing these, you're missing M1, you've got T1 and P1 that help out. Okay. So remember there's examples of these non-synonymous substitutions. We talked about what all these substitutions were, the definitions of them yesterday. Splice site mutations have been noted, and these are all the enzymes that are used in pharmacogenomics. So a lot of these are used to process drugs. Although, I mean, we don't have them because drugs weren't around. They were there around to process poisons in our environment. But you can see that all of these types of, the point of this slide is that all of these types of mutations have been discovered in different enzymes that are used in drug metabolism. Okay. And change promoter function, CYP2A5. Okay. And so when we talk about untranslated regions, we can have the 5' untranslated region of a message and the 3' untranslated region. There are promoters that are present really close by the gene. Do you remember what the name is of a sequence that's located far away from the gene, but that it still affects gene transcription? We talk a lot about promoters, but what is the other sequence of DNA that works? Enhancers. Yep, exactly. Okay. So, and remember the nomenclature of these different polymorphisms that have been described. So if I were to ask you about UGT 1A1 star 1, what does the star 1 mean? when I'm talking about any allele of any gene, star one. Typically, it's the most common. You're right, Tommy. Okay. So if you see anything other than star one notation on a gene, it means that it is a gene where a polymorphism has been detected in one population or another. It doesn't matter. Once it's reported, it's given a number, star 2, 3, 5, 7, up to 28 here. Star 128 decreases 1A1 expression. And that's because it has a 7 repeat variant, okay? So look at this. Star 28 is up to 30% of people that were analyzed for this study, okay? So if we have two different variants, they can have different effects. Remember this from enzyme kinetics? Oh, bringing back memories, right? So we talk about the common genetic form where you have the rate of metabolism here, and you have two different variants. This has an increased Km, and this one has a decreased Vmax. Remember how we measured those in lab? Anybody want to help out with lab this year? No? Can't talk you into it? I can talk you into it? Okay, we'll see what your schedule looks like. So when we have this, this is just a notation that a lot of times these variants can have different effects on drug metabolism. And then the other thing for you to remember about or to get exposed to that we never went over, I don't think, is haplotype blocks. So haplotype blocks are there for us to be able to interrogate the identity of a gene by only looking at one polymorphism. What has been done is that this sequence of the gene has been, this section of the gene has been sequenced many, many times. And what has been found is that in this haplotype block, you could interrogate, you could ask which variant is inherited of any one of these 1, 2, 3, 4, 5, 6, 7, any of these 7 polymorphisms. So these RS sequences are identified polymorphisms that are present in a database. And if I wanted to know what the most likely sequence of this whole section of DNA is, all I would have to do is to find out which form of the polymorphism is present in that first one. And since these are all tightly inherited close together, I would pretty much know what the rest of those sequences are. And that's where what we do is to find out, to more quickly figure out sometimes what is inherited by just doing 1 or 2 or 3 interrogations instead of resequencing that entire section of exon 1 through exon 5 of this gene. Okay. Just because, and I want you to know this just because, once again, it's vocabulary. So when you're out on rotations or you're doing maybe a research rotation or something like that, and somebody talks to you about a haplotype, you know that a haplotype is the set of alleles that are inherited similarly from one section of a chromosome. Okay. So the consequences of variation. This is a picture of the DAT dopamine transporter. And this is showing you the polymorphisms that have been identified. So one of the things I want you to notice is that these are all, these polymorphisms are all synonymous variants, okay? So these, remember what synonymous variants are? Who wants to answer? Yes. Correct. So these mean that there's no amino acid change. And you can see that some are in the intracellular space, some are in the extracellular space, and some are within the membrane itself. But they're not changing any sequence of the amino acids. Whereas here, if we're looking at a multi-drug resistance protein 2, MRP2, The red circles are non-synonymous changes. And you can see how many have been identified in this set of 100 African Americans, 100 European Americans, 30 Asians, 10 Mexicans, and 7 Pacific Islanders were part of this group. So you can see that apparently a lot more variation can be tolerated with MRP2 than could be tolerated with DAT, because we didn't see any non-synonymous changes in DAT. Okay. So what do you think could be the consequences then of, say, in this cytosolic region of some of these non-synonymous changes? Right. Do you remember what multi-drug resistance proteins do? What do they do? Yeah, you're working really hard to get that drug in the cell and to get it metabolized and out to where it's supposed to be. You know, say if it's a prodrug, it's got to get to its active metabolite and get it out, or it's got to be metabolized so you don't OD your patient. But if you're fighting against this gene that can be upregulated, its synthesis can be upregulated. That protein is just fighting to pump that nice drug that you designed so carefully to get in there and get it out. So when we're talking about different traits, we're going to talk about monogenic versus multigenic. So a monogenic means that it only has one gene involved, right? The opposite of multifactorial, right? In which we have multiple genes and multiple environmental factors. Now we're talking about a single trait. And if you have 2 alleles, one with low activity and one with high activity, now you can expect in the population to have a different amount of enzyme activity based on the phenotype. So if you have a homozygote for the high activity allele 1B, now you're going to have a whole lot of high activity and a little bit more frequency in the population typically. It's a lot different when you deal with reality, and reality is that multiple enzymes are typically responsible for any kind of pathway. We saw that before. We'll look at it again in a few slides. So in this case, in a multigenic trait, and you're looking at all these possible alleles for these 5 different genes, you can see that you can have a multiplicity of effects, and you're not going to be able to really ascribe it to any one because each of these is contributing. So it's going to change the risk for thrombosis in this case with the population frequency. All right. So there's a lot of databases that are available to you. PharmGKB is one of the most common ones. It's the one that everybody is using nowadays. You'll probably hear about it a lot in PK from Erica. And then DB-SNP used to be the biggest one, but the National Library of Medicine is losing a lot of funding, so we're not keeping up with it quite as well. But the most important one, PharmGKB, really is important for you guys to as a resource for you, because it's giving you genotype and phenotype data that are related to drugs. So when you're looking for things, that's an important place for you guys to look. Okay. And then this is just, like I told you last year, I will never ask you anything that's available on a chart because you can look it up just like I can, right? So, but the whole point of showing you this is that you have different types of variants that we've all talked about. And then where it occurs, whether it's in the coding region or a non-coding region, promoter region, regulatory regions, and then what is the functional effect. So this chart is just to point out that these effects are real, okay? The people have real results from inheriting different alleles, okay? And then this is to show you a functional effect. So this is MPP uptake is a way of detecting proliferation. Or I mean, of detecting rather the transporter efficiency. And you can see that the wild type or the reference, we're going to set arbitrarily at 100%. And then moving forward, you can see that the point of this graph is to show you that you can have nearly, pretty much no effect from a variant. You can have a lot of effect from a variant where you have, in this case, this variant will give you 200%, so twice the amount of uptake. And some of those will actually obliterate any transport possibilities compared to a water control. So all this is showing you is that it really depends on the allele that you're looking at as to what the effect is you see in the patient. Okay, any questions about any of that? All right. So when we're looking at the types of variants that we find, it makes sense that we see the most variants are present either in between the genes or in introns. Because if those have the least important effect, then this is really the ones that could be inherited most easily. If you inherit a deleterious allele within the gene, or even within a regulatory region, you're not going, that next generation is not going to survive. Okay. So it makes sense that you're going to find the most variants in the regions that appear to be the less important for the sequence of the protein that you're looking at. Okay. Doesn't mean that intronic SNPs don't have effects. We know that they do. So if you look at CYP3A1 star 1 and you have 13 exons, you put them all together and you're going to get an active enzyme. But in the case of the star 3 allele, there's an intronic SNP that creates an alternative splice site. When you put that alternative exon in, you now have a 3B in between 3 and 4. You're changing the reading frame. You end up with an early stop codon. So you end up with an inactive smaller protein. Does that help visualize it a little bit better? Okay. Think about it. You're essentially doing a huge indel here and changing that reading frame. Okay. So if we're looking at CYP3A5, and so you've seen the two alleles, right? The active one and then the truncated one that is inactive. And on the y-axis, we're looking at hepatic CYP3A5 mediated hydroxylation of midazolam. If you have you're homozygous for star 1, you're going to have a lot of activity. And if you're a heterozygote for star 1 and star 3, you're going to have an intermediate activity. Likewise, if you're a homozygote for star 3, you're going to have minimal activity compared to what you would need. Okay. So once again, if you look at another drug, you're looking at tacrolimus concentrations. If you have both star 1, you're going to have a what is considered to be the normal amount of tacrolimus. You're going to process this drug properly so that the usual dose is going to be fine for this patient. When you have a heterozygote star 1, star 3, now you're getting a little bit more tacrolimus hanging around. It's not getting processed as quickly. And if you have your patient is a star 3 homozygote, you're overdosing that patient. Okay. And similarly, we can go through the same kind of process of thinking of CYP2C19. Okay. So area under the curve, you guys are all getting used to area under the curves, right? In MedChem. And then intergenic pH. So what is it? this is just showing you that this has multiple effects, and whenever you're looking at a homozygous extensive metabolizer, a heterozygous extensive metabolizer versus a poor metabolizer, that's what these letters mean, you are going to see this type of effect. Okay. So when we talk about phase 1 enzymes, the majority, I think I showed you this last year, the majority of those enzymes or of the drugs are metabolized by CYP3A4, CYP3A5, a very similar activity, okay? The next most common one is CYP2D6. You can see it has a bigger chunk of the pie. 2C9 is in here too. 2C8 and 9 has another huge chunk. And then, so as we said, phase 1 enzymes metabolize drugs to be either, if it's a prodrug, it metabolizes it to the active form. If it's a drug, it metabolizes it into inactive or less active metabolites. And phase 2 is conjugating those drugs so that they can be eliminated. So I asked you to tell me what the other enzyme besides VKORC1 was important for warfarin. Anybody look it up? Anybody look ahead to the next slide? Yeah, 2C9. So there are two ways that this affects how warfarin works. You have CYP2C9 is taking warfarin, which is the active form, and making it hydroxywarfarin, which is less active or inactive. And then at the same time, you have VKORC1, which is responsible for making the most potent form of vitamin K. And vitamin K is important for clotting, right? Because what it does is it helps process antithrombotic factors into prothrombic factors. So when you're giving somebody warfarin, you're trying to prevent them from clotting. Okay, they have some condition, AFib, you name it, where they're in danger of a stroke from having clots thrown. Okay, so a stroke in the heart or in the brain, or a stroke of an MI in the heart. Anytime you get to a small capillary bed that can be affected. So it's a balance, right, between the vitamin K receptor kinase, and not kinase, vitamin K, I can't remember now, ORC1, anybody remember? I don't remember either. So, but the point is that it's the balance between those two enzymes that's important in regulating how much warfarin is available. So once again, this is useful to genotype people if they're in a rural area, they cannot get in to get typed, they cannot get in to get their INRs done. And what happens is if the usual dose of, say, you want recommended starting dose for a homozygote is at 5 mg per day. If you give somebody 5 mg per day who can't process it properly, you're going to overdose them on warfarin, they're going to bleed out. The whole problem is warfarin's a great drug and it's very cheap, which is why it's attractive, but it has a very narrow therapeutic index. So it's really easy to give somebody too much or too little. So when we have, if you have somebody that's going to an anticoagulation clinic, when they're first put on warfarin, they're going to go a couple of times a week to have their INRs tested and have their dose adjusted. And then they may go once a week, and then they go once a month. The problem is that adjustment time. And if you have a patient in a rural area who is working and has to travel to a clinic to get their doses adjusted, they may not have the ability to do that very easily, or it may cost them time away from their job or time away from their ranch or they don't have transport easily or whatever. If you can get them started closer to the best dose for them by genotyping them, then that makes life a whole lot, getting to the right INR is a whole lot faster. Okay. Now, there are other drugs now on the market that don't do this, but they're a whole lot more expensive. And that's why warfarin is still around. Okay. So another good example I think I've talked to you about is CYP2D6 with codeine. So codeine is not the active form of the drug. The active form of the drug is morphine. So why don't we give people morphine? Why don't we just avoid this altogether? It's more easily abused, so it is dangerous in that way. They're all cheap at this point. It's mainly because it is so easily abused. Okay. So if you have missing a copy of 2D6, then you're not going to process that codeine. You're going to be like my mom. She could eat codeine till the cows came home, and she had zero pain relief for her arthritis from it. So what did the doctor do? He prescribed her a different drug, and guess what? It goes through the same pathway with CYP2D6. And I'm calling my sister and saying, can you just talk to the doctor and tell him to order a different drug that doesn't use that pathway? And at the time I had Cheryl Brown tell me, okay, what are the alternatives that I can tell my sister to tell the doctor to tell my mom, give my mom, you know? But my mom was one of those people that said, well, I figured, well, it didn't work before, but maybe it works now. I'm like, you're getting older. Your enzymes are getting less active, not more active. Let's get you something that'll work. So what we're trying to do is look at absorbance and clearance of those therapeutic agents, trying to figure out the best efficacy, reducing the adverse reactions by making this much more personalized. And I'm going to skip ahead because I wanted to talk to you about a really big, this cancer treatment with 5-FU. How many of you are familiar with 5-FU? Used to be a very common cancer treatment, okay? So the problem with polymorphisms in the genes that take care of 5-FU is if they metabolize the drug really quickly, it doesn't have enough time to interact and kill the cells. And if you metabolize the drug very slowly, then you end up killing your liver or killing your patient's liver. So the reason I wanted to bring this example to you guys and emphasize it is because of this. So this is the normal case. When 5-FU is here, the majority of the drug is inactive because it interacts with DPD, okay? So you're giving a dose, a tolerable dose, and 15% of it, the drug is active, will interfere with the thymidine synthase enzyme, right? So thymidine synthase, what happens if you interfere with thymidine synthase? What do you use thymidine for? What's it a component of in your body? DNA, okay? So if you're inhibiting thymidine synthase, you're inhibiting the ability of those cells to reproduce. So it's going to attack rapidly reproducing cells, typically cancer cells. I mean, it also has side effects that your hair cells are growing, your skin is growing, the lining of your gut is growing, and that's why you have the side effects from this kind of drug. But the effective therapeutic dose is going to hopefully kill the cancer cells. It'll also kill some of the healthy cells, which you see the side effects happening. But in effect, this is going to work to reduce the cancer burden on that patient. Now look at the situation where if you have a DPD deficiency, now you're having a whole lot more dose. So even though you gave your patient the same dose, you're, instead of having only 15% of the drug being active, now you've got up to 100% of the drug being active. You're overdosing the patient. You're going to have way more drug. You're going to absolutely inhibit all of the thymidine synthase that's around, and you're going to kill everybody. You're going to kill healthy cells and the cancer cells too. And you're going to end up killing your patient. Okay. The other thing is if you have a decreased level of the thymine synthase enzyme because the person has a polymorphism where they have lower levels here. Look at, like your 3 little blobs of enzyme versus the 9. So now there's not enough around. So once again, you're going to have a whole lot of, you don't have enough thymine synthase around. You've killed what's there. So once again, you're going to kill all the cells. They're not going to be able to replicate. Okay. So you end up with toxic doses when neither of those are. So one of our faculty members had cancer and they wanted to put him on 5-FU. And I kind of gently freaked out. I didn't freak out. I just said, could you suggest to your doctor that they might want to look at the genetics in case this is a problem? We don't want to kill the liver of this patient. And they did. And because of the ethnic background of that patient, they said, well, listen, the chances are that this is not going to affect them that greatly. And this is a very effective drug for their type of cancer. So we're going to monitor them very carefully, monitor their liver enzymes, make sure that the liver's still working properly, and go ahead. And actually, he was able to tolerate the drug fine, and he's cancer-free now for 8 years. So we're calling it good. All right. So the whole idea of pharmacogenetics is to be able to tailor eventually that therapeutic regimen to the patient for the best outcome possible with the least amount of side effects and the best efficacy. Okay. Bottom line, if you wanted to break it down to, don't tell Erica I said that, but if you wanted to break it down to what is the most important from your perspective, that's probably it. Okay. Any questions? All right, you have study guides from yesterday and today. If you have any questions, go ahead and shoot me an email or find me in the hallway. You guys, oh, and you know where I live, except that I'm moving. So I'm moving from my office at the south end of the building up to next to Erica. So I'll be up here in 341. Okay. All right. Enjoy the rest of your day.